All right, good morning. I'm Shelley Miller, president of the conference, and we've had a great start to the day. We had an early morning hike with students. Here we are. 18 of them showed up. It was great, and we hiked Mount Sanitas. Beautiful morning. I'm really excited to introduce our first plenary speaker, Dr. Arlene Blum. She is a biochemist, biophysical chemist, author, and mountaineer, and is visiting scholar in the chemistry department at the University of California, Berkeley. She's the executive director of the Greene Science Policy Institute. And there's some information about this institute, and signing up for the newsletter is an excellent idea. I love it when I get it every month. It's excellent. So I encourage you to do the same. The institute brings government, industry, scientists, and citizens together worldwide to support chemical policies to protect human health and the global environment. Her research and policy work to protect children and humans have been preventing the use of billions of pounds of harmful flame retardant chemicals in children's sleepwear, furniture, electronics, and other products worldwide. I met Arlene when she was here for the Conference on World Affairs, and she gave a talk to my indoor air class, and it was the first I'd heard on flame retardants, and since then, luckily, they've been banned from furniture in California. Almost. She's going to clear that up for you. One of the things that I wanted to point to you is that Arlene Blum led the first American and all-women's ascent of Annapurna. Considered one of the world's most dangerous and difficult mountains, co-led the first women's team to climb Denali and completed the Great Himalayan Traverse across the mountain ranges of Bhutan, Nepal, and India. Very accomplished mountaineer. And if you are looking for an exciting book, you should read Annapurna, A Woman's Place, and Breaking Trail Climbing Life. They're really excellent. So I'm going to turn the floor over now to Dr. Blum.
Well, it's great to be here in Colorado. As a mountain climber, I really appreciate it. Now, let's see, how do we start my talk? Great. Okay, good. So what I'm going to talk today is about how scientific research, when you bring it to decision makers, can change policy, and it really can. So the things you know, decision makers don't know, they often get their information from people with a vested interest in the outcome. And if impartial scientists and engineers share what they know, they're better decision-making outcomes. But what I've learned most recently is by impacting purchasing, big purchasing, we can have an easier and quicker impact than policy, which is really slow and difficult. So let's see. So I started doing chemistry and mountain climbing at Reed College in the 60s. My freshman chemistry professor was 23 and had a PhD from MIT. There were 4 girls in my chemistry class. We all got PhDs in chemistry. At the same time, there were all those handsome young mountaineers around, so I started climbing. And I kept alternating chemistry and climbing in my career. And way back in the mid-70s, my climbing partner, a brilliant young climber, died in India. And I really wanted to do something for the environment. Bruce Ames, who some of you may have heard of, a biochemist at Berkeley, who had the Ames test, had been on my orals committee at Berkeley. And I said to Bruce, what can I do for the environment, in the memory of my friend? And he said, well, why don't you look at the flame retardants in children's sleepwear? And that seemed like a really peculiar thing to me. And he said, yeah, it's so applied that even the undergraduates won't work on it. But I was depressed because my friend had died, so I thought I'd work on flame retardant. So it really sounded strange. So Bruce said there was a flame retardant called brominated tris that was 10% of the weight of children's sleepwear. He thought it got into the children. He thought it was harmful. So we found a little girl whose mom had bought her sleepwear in England, put her in the tris sleepwear, and the next morning there were tris breakdown products in her urine. So in America, foods, drugs, pesticides, things that that we eat in one way or another are regulated, but other chemicals really weren't. And here was an unregulated chemical that was ending up in children. So we ran a mutagenicity screen, and tris turned out to be one of the strongest mutagens we'd ever seen, a likely carcinogen. Most all the kids in America were wearing pajamas that were 10% tris, and we knew it got into the children. Do any of you remember that story? So I thought, oh my goodness, we have to write a paper, we have to warn the families in America that they shouldn't wear tris pajamas, and then I get invited to climb this mountain. Do you know what mountain it is? It's Everest. And at that time, no American woman had ever tried Everest. So I had this terrible decision. Did I stay home and warn everybody about the tris pajamas, or did I go climb Mount Everest? So let me ask, this is a Colorado crowd, how many of you would climb Mount Everest? How many of you would do your paper on the tris pajamas? Okay, well, I actually did both. So I went to Everest, climbed through the Khumbu icefall, strung ladders across the big crevasses, went up steep ice faces, and every night I got to camp and I wrote more of my paper. So I got to my high point, that's 24.5, and my flag showed the Petri dish. Let's see. Yes, that's, pointer isn't working. The Petri dish on the left has no tris, there are only 2 colonies, but the one on the right, it's blowing the wind, had tris, so there were a lot of colonies. So that was my high point, 24.5. I sent my manuscript by mail runner back to Kathmandu. It went to Berkeley, and it was published in Science about 3 months later. And the subtitle says a lot about our institute and what we do. It says the main flame retardant in children's pajamas is a mutagen and should not be used. So now, what do most all scientific papers recommend at the end? More research. And here we just done a couple experiments, wrote a paper with a really strong recommendation. That was January 1977. And 3 months later, tris was banned from kids' pajamas. And that's the way things should be, right? We share the science, things change. But of course, when brominated tris was banned, industry always wants to keep things as similar as possible, so what's the replacement? Chlorinated tris. So we did some more studies. It was a mutagen. We wrote another paper in Science. Chlorinated tris was removed from kids' pajamas, and I thought everything was fine. But what I didn't know is that chlorinated tris continued to be used. Anybody have a couch they bought since 2006? So you have about 2/3 chance you have tris in your couch, and 1/3 chance of something worse. And the reason is it goes back to the 70s also, we do not have protection against harmful chemicals. Do people know this? Does anybody here think that if a chemical's in our toothpaste or our shampoo that the government's protecting us? We all know that it's not, but most people think it is. And the only law we have to protect us against harmful chemicals before they go out in the world was TSCA from 1976, and when it passed, 62,000 chemicals were grandfathered, and they really can't be touched. Asbestos is one of them. The EPA spent years trying to regulate asbestos. They did not succeed. The standard of proof is so high that asbestos cannot be regulated. And you're saying, wait, we can't use asbestos, there are lawsuits. Right, there are lawsuits. And people who have mesothelioma and are asbestos workers, because asbestos is the only cause of mesothelioma, they sue, they win, they die. Everybody with mesothelioma dies. So this is not a good way to regulate chemicals. So old chemicals like tris, the EPA doesn't even try to regulate them. New chemicals, there's no requirement for any health data, any data at all. 2/3 have no data at all. So we are in a place where we're really not protected against chemicals. And 2 brilliant pediatricians said we are conducting a massive clinical toxicological trial, and our children and our children's children are the experimental subjects. We are the experimental subjects. All these chemicals are going out into the world, ending up in us, our children, our environment, our wildlife, with no testing, no regulation. The other problem is when you regulate or phase out a chemical, regrettable substitutions. So on the left, you have decabrominated diphenyl ether. It's a flame retardant that's used in a lot of plastic, fabric back coatings. It's very persistent, bioaccumulative, and toxic. After many years' effort, it was phased out. And what's the replacement? Decabrominated diphenyl ethane. which also is just as maybe more persistent and bioaccumulative. We don't yet know about the toxicity. So the ether sales go up, the ethane go down, the ethane sales go up. Not much benefit. So a group of us who have been working in this area got together at a series of retreats, and it was people from government, academia, NGOs, and business, and said, can we figure out some way to tackle this problem? Because when you hear 80,000 untested chemicals, what do you do? And so a lot of chemists at this meeting, maybe 2/3, and we said, well, why don't we think about classes of chemicals that contain most of the harmful classes? And of course, the halogens are an obvious choice. When you have fluorine, chlorine, or bromine bonded to carbon, it's a strong bond. There's no naturally occurring organohalogens in any mammalian systems, so our bodies really don't know what to do with them. The things that keep toxics out of our cells don't keep them out. The things that clean out our cells don't clean them out. So they tend to go into our bodies, stick around, and cause trouble. So we had a retreat, and we thought about what are classes or families of chemicals where there's irrefutable evidence, thousands of peer-reviewed papers showing harm. And we came up with 6 classes. And I'm going to tell you about them today. They're the highly fluorinated chemicals, stain and water repellents. I came here because there was a meeting of 300 scientists at the School of Mines last weekend who are studying highly fluorinated chemicals. Chlorinated antimicrobials, flame retardants, bisphenols and phthalates, organic solvents, and certain metals. And there are handouts as you leave on both sides that list all the classes and give you some information about them and how to learn more. So when we decided, things really had to reach a pretty high standard. We were worried about nano, but there wasn't that much data. We were worried about siloxanes, there wasn't that much data. These had to be things that had a really high standard of proof. And the point was, when you regulate or phase out one chemical in this class, you don't go to another one. You go somewhere else. And when things are in this class, you ask, is it necessary? So fluorinated chemicals give you great stain and water repellency. Maybe you need them to climb Mount Everest. Do you need them in your bathing suit? Is it worth it? If you say to a mom, well, you can have a nice stain repellent carpet, but the chemical is going to be in your baby's body for the next 20 years and in the environment for the next 1,000,000 years, is it worth it? Then if you think it's necessary, is there a safer alternative? And if there isn't, then really invest in green chemistry. Green chemistry is a chemistry usually not based on petroleum, based on renewables, and it's the design of chemicals that reduce the use of hazardous substances. It's been around for more than 2 decades. Very little resources have gone into it. It's time to really resource it and use it, particularly to find replacements for these 6 classes. We have a series of webinars. The flyers out there tell you more. They're 15-minute webinars in each of the 6 classes where we explain the chemistry, why they're harmful, where they're used, what to do about it. And when we did the webinar series, 1,200 people listened, including people from most of the major companies in the United States, even around the world. We had told people, because people whose job it is for big companies to make decisions about chemicals often aren't chemists. They have big red lists and CAS numbers, and they want to do the right thing, but they don't understand it. It's really confusing. So we said, listen to this webinar. You'll understand from the inside why you don't want to use these chemicals. And amazing things happened. The day after the antimicrobial lecture, we got a call from Target saying, we had no idea we had triclosan in our home line of personal care products, and we're taking it out, and they did. So companies don't have any reason to put toxic chemicals in their products. They used to get most of their information from the chemical industry, whose information was maybe not always the best. And when they get better information, and when things are in a package where they can understand it simply, they can make better choices. So we think purchasers are key. Manufacturers, retailers, big buyers, public procurers, and even consumers. So consumers are hard. We don't really work with them too much, but the others we do work with. So I'm going to start with flame retardants because this is where it started for me, and it started again in the 70s when rules were made saying that children's sleepwear, furniture, and foam plastic building insulation had to meet fire standards. And they were open flame standards, so the only way really to meet them was by putting flame retardant chemicals in these products. Nobody said, do the flame retardants provide a benefit? Nobody said, are they harmful? And we've come to learn that in most cases there was no benefit and serious harm, but huge profit. And so that is the story of flame retardants. And banning flame retardants does not work because there's always the chemical cousin, right? Brominated tris, chlorinated tris, the redoubtable substitution. So if any of you, pretty much, if you live in North America, go home and pick up the center cushion on your couch, you'll see a label saying it means Technical Bulletin 117, and that means 5% of the weight of the foam is tris or firemaster or something worse. And there's no significant fire safety benefit. The standard says the furniture foam won't burn for 12 seconds when exposed to a very small flame. If it's a larger flame, it burns right away. If you drop a candle on your couch, what burns first? The fabric. You have a larger flame. The foam burns almost immediately, and in the presence of flame retardants, there's a lot more soot, smoke, and toxic gases. So it's been well established, no fire safety benefit. The chemical that was used before 2005 is one of only 23 chemicals banned globally in 150 countries by the Stockholm Convention. Oops. Let's see, go back. It's called penta-5-brominated diphenyl ether. And it's halfway in structure between a PCB and a dioxin. So if you have a PCB, instead of a covalent bond, you have an ether bond, you have a diphenyl ether. If you burn or oxidize, you get a substantial amount of dioxin. So that molecule is 5% of the weight of the foam in most furniture from before 2005. It was used from 1975 to 2004. 98% of the use was here in foam, and it has become a huge global pollutant. There's a whole industry of scientists studying it in wildlife and the environment and humans all over the world. These chemicals migrate. There's literally thousands of papers, many hundreds of animal studies now because it's been used so long. They're human health studies. Human health studies are not a good thing. By the time you have human health studies, you're in trouble. Women with higher levels of penta take longer to get pregnant, have altered thyroid hormones during critical windows of brain development during pregnancy, so their children have lower birth weight, impaired attention, poor coordination, lowered IQ. It's been estimated the average California child has about 5 less IQ points due to penta, which is in almost all of us, particularly those of us in California. The chemicals are SVOCs. They migrate out into dust. Most of our exposure is through hand-to-mouth contact, having dust on your hand, eating a sandwich. Children crawl in the dust and sadly have 3 to 5 times higher levels of these flame retardants in their bodies than their parents because of the hand-to-mouth exposure, just at the time of developing reproductive organs and brain development. And of course, cats have 10 to 100 times higher behavior than humans. Why would that be? Grooming behavior. Look, they get it on their fur, they lick their fur. As we said, no fire safety benefit, no difference whether you have the chemicals or don't have the chemicals. So way back when I started doing this, I remembered the 70s, somebody said, let's write a bill. And I actually was going to, my daughter had just started college, I was going to move to Nepal and walk across the Himalayas again. And I said, okay, I'll stay here. That was 2006. I said, okay, I'll stay in America a few months. I'll help write a bill. We'll get the flame retardants out of the furniture. And so there were 4 bills. And they all would have increased fire safety without the use of these harmful flame retardants. And they were, the furniture, foam, and fabric industry all supported it. They didn't like exposing their workers or the public to the chemicals. They knew there was no benefit. Firefighters supported it because they have high levels of cancer associated with dioxin exposure. And when these burn, you get a lot of brominated dioxins. citizens organ, everybody supported it. There'd be a hearing with 40 firefighters, business people, parents, scientists, saying we support the bill. On the other side, there'd be 3 lobbyists from the chemical industry, and you know who our fine representatives would vote for. So that went on for about 6 years. Once we almost passed, and this is a $6 million one-week advertising campaign from Californians for Fire Safety. Those are the 3 companies that make the flame retardants. So this is kind of the reality of when you change regulations so you don't need harmful chemicals, you are right up against the chemical industry. When you get purchasers to stop buying them, it just happens overnight. In any case, this went on for about 6 years. Along the way, I got tired of going to hearings where the lobbyists smoothly said there's no good science showing any harm from flame retardants. So at a dioxin meeting in San Antonio, we wrote the San Antonio Statement on showing the harm, lack of fire safety benefit of the chemicals, hundreds of references signed by 200 scientists. So we really had a document. And for us, that's been step 1 with classes of chemicals, is get a consensus statement from the leading scientists all over the world, and it's pretty irrefutable when you do. Shortly after that, nobody pays that much attention to scientific consensus statements, but they're good to have. The Chicago Tribune came out with a series with similar information, a little more scandalous, and the subtitle says it all. A deceptive campaign by industry brought toxic flame retardants into our homes and our bodies, and the chemicals don't even work as promised. And it was a great series. It came in second for the Pulitzer Prize, won all these environmental journalism awards, and it changed everything. Before this series, when I go to a party, people would ask me what I do, and I'd start telling them about the toxic flame retardants and how they had no benefit, and they'd always say, I have to leave now, and I'd see them talking to people on the other side of the room. And I thought I would spend the rest of my life, getting increasingly this kind of wacky lady who just rants. It's like the Emperor's New Clothes. When you see something, you have to tell people. But after the Tribune series came out, Dick Durbin, who was the majority whip in the Senate, started ranting just like I did, and he had a lot more impact. There were hearings in the Senate, and our governor, I started hiking with Jerry Brown's senior advisor, so he got informed, the governor got informed, and the governor said, we must find better ways to meet fire standards by reducing and eliminating dangerous chemicals, told the agency in charge to change the standard. No legislature. So it took a while. That was 2012. We meanwhile got an EPA grant to work with the trade associations for furniture, foam, fabric, juvenile products, really get them educated, fully informed. And January 1st, flame retardants are not needed in furniture. They can be used, they're not needed. However, if you go buy furniture, if it's manufactured since January 1st, it's going to have a tag, and it's going to say contains added flame retardants or contains no added flame retardants. It says lots of bad things about flame retardants. So this is good news for you. If any of you don't want your toxic couch anymore, you can go get another one. Then the next problem is what happens to all the toxic couches out there? There are lots of them. What are you going to do with your couch? And the answers are low-income communities or student housing, not so great. Landfill, there's a fair number of studies that they eventually leach out. The foam gets chopped up and put into bonded carpet cushion. You go to Home Depot and buy some foam and measure it, it has flame retardants, some of which are even the old banned ones in there, because it's below the levels at which they're banned. So we are working on answers. We have funding from NSF and NIHS for a meeting in November on what do we do with foam and plastic mixed with flame retardants. And if any of you are interested in this subject, be sure to see me, because we're wanting to bring together scientists to develop new separation technologies. Maybe we can separate out the flame retardants from the foam, remake the foam, and then what do you do with molecules that are so harmful once you've created them? So anyway, we're just starting to invite people to this meeting, but let me know if you think you're interested. Okay, what's probably really interesting to you, if you're interested in healthy buildings, is, remember, in the 70s, foam plastic insulation, that's polystyrene, polyurethane, polyiso, which are increasingly used for energy efficiency, all have to meet the Steiner tunnel test, which is met with the worst flame retardants. So you have a super green platinum building full of polystyrene, and you have huge amounts of hexabromocyclododecane, bad chemical. And the foam with flame retardants is used inside walls, below grades, attics. Again, that's our thing, we wrote a paper. This long paper, we had 14 pages of reviewers' comments, which we eventually answered, got it published, discussing the harm and lack of benefit of the flame retardants in building insulation. The flame retardant used in polyurethane and polyiso is a form of tris, similar to what was in kids' pajamas with health problems. The one used in polystyrene, XPS, and EPS is HBCD. It's bioaccumulative, persistent, toxic, banned in 150 countries under the Stockholm Convention. It is being phased out. What do you think the replacement is? You think it's going to be good? Poly-FR, brominated butadiene styrene copolymer. Butadiene's cancer-causing, styrene's cancer-causing. You put them together, make a polymer, and brominate. Does the planet need a lot of that? And our three companies, the same ones who used to be called Californians for Fire Safety, they are all making it under great names like Emerald Innovation, Greencrest. I mean, who could not like those chemicals, right? And the use is going way up because now in California, wall cavities have to be twice as thick, twice as much insulation, twice as much Emerald Innovation. So substitution is not the answer. We are working to change the International Building Code. That is the answer. And we started with just insulation below grade, where there's no fuel source, there's no oxygen, so no fire, and we are trying to change the code. I have heard that someone said it's a very high priority of the American Chemical Council that we not be successful. It's been really hard. We'd hear it's a no-brainer, and then suddenly people would vote against it. It's been a real challenge. We've heard great arguments from the chemical industry. What if there's an earthquake, the foundation splits, somebody pulls out that insulation and uses it in an attic? They seriously say things like that. But during our first round at the ICC, it did not pass. We got a second chance in September. Avery at Green Science Policy is in charge of this incredibly difficult project. If anyone's interested in helping, contact her or me. A platinum building that's full of Emerald Innovation may not really be the healthiest building. The good news is in Scandinavia, they don't use any of these chemicals in their insulation, and they've had no increase in fires. But the voting committees do not listen to this as much as they might. Okay, I'm going to try to go quick. How much time do I have? 5 minutes. Okay. Chlorinated chemicals. The carbon fluorine is the strongest bond in nature. So these molecules really have unique oil and water repellency, but they don't break down for geologic time. That could be longer than mankind. So that's PFOA, C8, 8 carbons with fluorines around. A favorite book, Stain-Resistant, Nonstick, Waterproof, and Lethal, the story of C8. Where do you find it? In all these sorts of products, food contact papers, cosmetics, outdoor gear, fry pans. Health problems, there's a whole range of them because they've been leaked. DuPont plants making Teflon leaked them into the water supply in southern Ohio. Hundreds of thousands of people were exposed. This is human humans. So the replacement is C6, which also lasts for geologic time, and there are 40 kinds of C6. So we're never going to know about them all. The chemical industry assures us they're fine, but they're persistent. They're going up in groundwater, wastewater, seawater, human blood. Probably not the best. So we put together a Madrid statement. It was published in Environmental Health Perspectives May 1st. Again, signed by several hundred scientists. It's been getting a lot of publicity, which is great, and the headlines are 230 scientists say there's harm, chemical industry says there's not. So there really is power in bringing scientists together to make consensus statements. All these brands are eliminating fluorinated chemicals, not because of us, but because they were picketed by Greenpeace, but it worked. And we are working with big purchasers, giving them designers lists of products with and without fluorinated chemicals so they can make better choices. And if anybody comes to I have a 2-hour workshop where we're going to talk about action statements, practical things one can do to have healthier buildings by reducing the use of these classes. Antimicrobials, they look like dioxins. 1974, the FDA draft monograph on their lack of efficacy and safety has not yet been completed. Meanwhile, they were first detected in fish. There used to only be 7 dozen, but then they were no longer in the drug category. So there are now 2,000 antimicrobial products, top 10 water contaminant, endocrine disruptor in basically everybody's women's breast milk. So this is a class where there's really very little benefit. And right now, almost all building materials I've heard have Microban or triclosan, doorknobs, counters, but they also don't. So again, we're putting together a list for designers that do and don't, encouraging big purchasers to use the products without and move the market away from products with to products without. So I guess I'm really fast. Bisphenols and phthalates, organic solvents, certain metals. You can learn more at the workshop. But the big success, IKEA we've been working with for years, they have phased out all highly fluorinated chemicals from all their products worldwide. And DuPont had said they couldn't ever come up with anything better than C6, but shortly, now that IKEA says we're not buying any fluorinated chemicals, their alternative, which they say is renewable, comes from DuPont. So they actually did have a way of doing it. It might have been not as profitable, more costly, but they're doing it. They have aqueous stain resistance in the alternative. They don't have oil resistance. So IKEA said, well, we have these nice tablecloths and we need oil resistance, and we've given them 1 more year. And if they can come up with an alternative, we'll buy it, but if not, we're going to stop selling our tablecloths because we're not going to sell anything with highly fluorinated chemicals. And they have banned things in all the 6 classes. So the thing I'm really excited about is the Buyers Club. The USGBC, Northern California, has a building health initiative, and they put together a Buyers Club of these really large companies, and they are putting their tens of billions of dollars of purchasing power together, and they're starting with buildings. 1 minute, I'm almost done. To require transparency for manufacturers, what's in the products, and to create a demand for healthy products. And we're actually working with them, introducing the classes. We're trying to encourage them to start with antimicrobials, which is an easy one where they really can move the market. Harvard wants to do a Buyers Club, but for universities in the East. A similar thing, they're big purchasers. And these 4-day retreats we have the people from the Buyers Club and Harvard. They all came to our last retreat, and they really want to use their purchasing power. So the challenge, I have said that this could be possible. I have no idea. But can the use of some, or better yet, all of the 6 classes containing chemicals of concern be reduced by 50% in 5 years, 90% in 10 years? And I really think it's possible. Just the do we need it question, is it worth it? If people understand, and these are big purchasers, retailers, manufacturers, they can make other choices. About a month ago, I got a call from one of America's largest retailers. He said, July 1st, we're not putting any more fluorinated chemicals or any more flame retardants in any of our products. It's confidential for now. I haven't told anyone who it is because they first have to get rid of their old stock. But after the call, I sat and cried for about 2 hours. I was so happy. So IKEA's doing it. One big American retailer's doing it. I asked Walmart if we could have a meeting, and we've worked with Walmart and all these retailers around flame retardants. I asked Walmart, can we have a meeting and invite all the retailers and let them hear what IKEA did? I don't know if it'll happen, but if American retailers decided to do this and all those big purchasers decided, I think we can really shift the market. So Again, if you want to join our e-list, give me your card or leave it out there. We send out monthly emails. And so I'm really optimistic. And this is something I think we'll see 5 years from now what happens. But you know, when I got the July 1st message, I couldn't believe it. It was so much faster than I ever expected that an American big retailer would do that. But you know, it's the right thing. And they don't make a profit selling toxic chemicals, and when they really understand it, they can do different things. So that's my optimistic, hopeful message. Thank you.
All right, our next keynote speaker is Professor Jessica Greene. Jessica is the Director of Biology and the Built Environment and Associate Professor in the Department of Biology, University of Oregon. Dr. Greene's research lab applies theoretical, computational, and empirical approach to study complex systems in a wide range of habitats, bodies, cities, forests, and oceans. Her talk is about next generation indoor environment quality. Let's welcome Jessica.
Hi. Can you guys hear me all right? Okay. I'm Jessica Greene. That was a completely motivating talk. It reminded me that I feel like I do my best work when I'm in motion, if I'm swimming or climbing a mountain, and I should do that more often. I'm sure that people that are based here in Boulder, Colorado do that. I'm going to tell you about some of my ideas that are based on my experiences as the director at the Biology and Built Environment Center based at the University of Oregon, and also my more recent experience as an entrepreneur where I'm the CTO of Phylogen based in San Francisco. I've been thinking about indoor environmental quality for a long time. Over 20 years ago, I was a graduate student at UC Berkeley, and I took courses from great professors, including Bill Nazaroff. And you can see that back then my name was Jessica Booher, and I'm sure you're not surprised that I changed my name when I married Steve Greene. Back then, when we were learning about indoor environmental quality, we learned about mass transport, and we learned about pollutants in the indoor environment and their fate and their sources. And much of what we learned was based on physics and chemistry. And there's a really good reason for that. We just learned about how important chemicals can be in the indoor environment. The very little that we learned about biology and microbiology at that time, we were taught that microbes were pollutants and something that should be removed from the indoor environment. And there were really good reasons for that. One of them was back then, one of the major ways that we tried to classify microbes was by looking at them under the microscope. And we now know that most microbes can't be differentiated when you look at them visually. And back then, I was a grad student in Lisa Alvarez-Cohen's lab. I spent a lot of time culturing microbes in the lab. And we now know that most microbes, if you bring them back into the lab, you can't culture them to find out everything about them that you would like to. If I was going to be a grad student today at Cal, I presume, this is what you would see if you were collecting environmental samples from any environment and wanted to learn what was going on in them. You would see DNA sequences made of the base pairs A, T, G, and C. And when I was a student at Cal, this great paper came out by Norman Pace, this was in 1997, that demonstrated, it was the first time that I had seen someone take these gene sequences, compare them to one another, and from them infer what different types of microbes were in those samples. So this is what you're looking at is a diagram of the tree of life, all of life on Earth, and most of the organisms that you see here are microscopic and can't be seen with the naked eye. So if you couple this way of comparing gene sequences to the exponential or more than exponential drop in the cost of DNA sequencing, combined, what that's done is it's transformed our understanding about a lot of environmental systems, but in particular, the human system. For every cell that is in the human body, there are at least 10 bacterial cells and probably 100 viruses. But through using DNA sequencing technology, we've learned that we're mostly microbe, and our microbes are really key to our health and well-being. There's a lot of great research going on in the human, about the human microbiome that's showing that the microbes in and on your body can influence how your immune system works. The microbes in and on your body can impact how resilient your body is to infection. And there is some research that's being done with model organisms that suggests that the microbes in and on our body may even play a role in how we think and how we feel. So the next obvious question, given that we know that microbes are important to our health, is to think about where do we pick our microbes up from? Given that we're going to spend 90% of our lives in the built environment, where we're regularly exchanging microbes with every breath that we take and every surface that we touch, the next obvious question after that is to think about, is it possible to manage the indoor environment so that it's beneficial to human health and well-being? I believe that we can do that. And I believe that based on my knowledge of what's been conducted in the scientific domain. I recently conducted a literature review of over 1,000 articles, all focused on the built environment microbiomes. That's a collection of viruses, bacteria, fungi in the built environment. A lot of these papers I found on a Mendeley group that's curated by MicrobeNet, which is funded by the Alfred P. Sloan Foundation. And if you look at these papers and many others, what you can see is every node here is a journal article, and the nodes are linked if they have similar words in the abstract or in the title. And then the major colors denote the themes of the different journal articles. And what you can see is that there's been a very rich body of literature focused on the indoor microbiome. And what these papers collectively tell you is, one, we know that indoor microbes impact human health. Hospitals are a great example of an indoor environment where we know that if you are going to go in a hospital, you are at a higher risk of contracting an infection than in any other environment that I know of. We also know that building design and operations impact the indoor microbiome. And I'm going to show you some data to demonstrate this. And there's many people in this room that are engaged in this research. And if you put these two lines together, what it suggests is that we can operate current buildings and design future ones to promote beneficial microbes and inhibit harmful ones. So what I want to do now is invite you all to think about what are the mechanisms that shape indoor microbiology. And I'm going to ask you to think about that on a conceptual level and how that relates to the way that we think about indoor environmental quality. So what do you think is the major source or a major source of microbes that enter buildings? That's right. Humans are a major vector of microbes into the built environment. Jordan Petchia's lab published a great paper, I hope I get the statistic right, Jordan, that every human on average contributes 40 million bacteria to the air within an hour. That's more bacteria than the number of people that live in the state of California. And we do this by directly shedding our microbes, but also resuspending microbes that are in the indoor environment. So all of you are sharing your microbes with one another now, and you are probably leaving behind some of your microbes in your chair, and they will be there for the next person that comes and sits down. We know that our pets that we bring into the indoor environment are also colonizing indoor spaces. Susan Lynch has published some great work from UCSF showing that if you take the dust in homes where people have dogs versus don't have dogs and you expose mice to that dust, you end up getting, with the dog dust, an enrichment of a strain of lactobacillus that improves the health and immune function of those mice. We know that microbes enter the indoor environment through windows, through mechanical ventilation systems, and also through all sources of water that come into the indoor environment. And once microbes are in the indoor environment, whether they thrive and survive and which taxa thrive and survive is a condition of what type of an environment they encounter. So some microbes do really well in a cold, dry environment, and others do well in a warm, moist environment. The types of materials that are in the indoor environment can serve as a habitat for different microbial communities. And finally, the way that we disturb the indoor environments, we just heard a great talk about this, is going to impact the types of microbes that are indoors. And of course, we disturb the indoor environment, for example, by using antimicrobials in our cleaning products, but they're also embedded in many of the materials that are in the indoor environment. Erica Hartman is here today. She's a postdoc at the BioBE Center who's leading some fantastic work looking at the influence of antimicrobials in the built environment. She's sitting next to Jeff Klein, who's also heavily engaged in this work. So next I'm going to show you some data to provide evidence that the way we design and operate buildings does impact the indoor microbiome. And of course, all of these different mechanisms that I just shared with you are ways are important for the indoor environment. I'm going to focus on ventilation and tell you 3 simple data stories that link ventilation to the indoor microbiome. But that's not to marginalize all the other ways that we can potentially influence the indoor environment. The first data story that I'm going to tell you is based on work from the Biology and Built Environment Center, where we looked at the influence of ventilation in hospital rooms. We did this work with Providence Health and Service